Good evening, everyone, and welcome to Shoals Marine Laboratory's weekly marine science seminar. I'm Dr. Jennifer Seavey, the Executive Director here at Shoals. For those of you who are not familiar with us, Shoals is the largest and the oldest undergraduate focused marine lab in the country and is jointly operated by the University of New Hampshire and Cornell. We are located in the Isles of Shoals, about 10 miles offshore of Portsmouth, New Hampshire. It has been an incredible day out here. Although we're located on Appledore, the Marine Lab, the students have been at sea and on the mainland studying salt marsh and sharks. And we've had a really full day. But every Thursday evening, we come together for these weekly rock talks, as we call them here on our rocky island. And we're excited to have all of you with us tonight over Zoom to join this community here. So our format tonight is 45-minute talk, followed by 15-minute Q&A. If you want to ask a question, which we hope that you will, please use the Q&A box at the bottom of your screen, type it in there, and then I will read them to our speaker at the end. If you have any technical needs or questions, please also use the Q&A and myself or Colin will help you out. So we're very excited to have a returning shoaler tonight on the island. Dr. Loretta Roberson actually taught underwater research with Jim Coyer out here just a little while ago, right? And she has her undergraduate in biology from Cal State University and a PhD in biological sciences from Stanford. In 2003, she became adjunct and later assistant professor at the University of Puerto Rico. And we've had a bunch of students from Puerto Rico on the island this summer, so that's very cool. And right now she is associate scientist at the Marine Biological Laboratory in Woods Hole. And her research interests focus on marine protection and marine conservation, which you're going to hear about tonight, and sustainable energy. So tonight she's going to talk about coral conservation and sustainability. And I'm really excited to have you with us here tonight, Loretta. So you can take it away.
Great. Thank you so much, Jen. I appreciate it. I'm so happy to be talking to you guys. Although I have to say, I am a little bit sad I can't be there at Appledore again with you all. Let me get this shared here. And okay. All right. So I thought what I would do is start by just talking a little bit about my path through my training and how I got to the research I'm doing now, just to kind of put things a little bit more into perspective. So Jen had mentioned I got my bachelor's degree and PhD in California. I'm a native to Southern California. And so that's where I learned how to scuba dive in the beautiful kelp forests off the Channel Islands and spent a lot of time at the USC Marine Lab out at Catalina Island. And this is me struggling with playing with electronics in seawater. Then later I moved to the University of Puerto Rico and was assistant professor in the Department of Environmental Science for a number of years there, and spent a lot of time at the UPR Marine Lab, which is at Isla Magueyes in La Parguera on the southern part of the island. And unfortunately, they don't have giant kelp there, so I had to kind of switch gears a little bit and started working on seagrasses, tropical seaweeds, and of course, corals. And this is me really enjoying the warm tropical water. And then about 5 years ago, I came here to Woods Hole at the MBL. And if you haven't been here before, we're kind of at the very bottom corner of Cape Cod. Not too far away. But diving here took a little bit of an adjustment after Puerto Rico, particularly certain times of the year, but we don't need to talk about that now during summer. But I was happy and pleased to see that we do have kelp here, which is awesome. And I thought I would just throw this image in just for yucks. On the right is sample of sugar kelp that was collected at Appledorn Island for a selective breeding program we have going on here at Hui. So, so I hope I can get back there sometime soon. But the amazing thing to me was that there's actually coral living here as well. And let me come back to and tell you more about this remarkable coral in a minute. But how I started working on corals and why we should work on corals is that I'm sure I don't have to tell this crowd, but we've seen an unprecedented amount of bleaching and damage to coral reefs worldwide due to increases in global temperature. And so what were once these really colorful, vibrant reefs look like they have been literally dumped in chlorine bleach. And this happens because the symbiosis that the coral has with the dinoflagellate that lives in its tissues breaks down due to high temperatures or some other stresses. And they leave the coral, and what's left is that skeleton. So I really wanted to understand why certain species bleach and others don't, and what are the consequences to reefs if this continues to happen. And so I just went back to the basics and tried to understand how corals function. And we know that reefs start with calcification. They have to make that skeleton framework for the reef that's made of calcium carbonate. And that space that's between the coral cells and the coral skeleton, we know certain ion exchange has to happen. We know calcium and carbonate obviously have to get there. But there's different pHs that have to happen. And surprisingly enough, we still don't know what all the key players are in the system and how they might respond to changes in temperature, changes in pH in the external seawater, et cetera. So the approach I took was to use different model systems to try and tease apart what's happening between the symbionts and the coral and that calcification process itself. And using a molecular approach as well to understand what genes get turned on when corals settle and begin to start making that skeleton. So right now we have the local coral Ostrangia, but we also have several tropical species, including Chiroptera, Porites, Monastria, and Aptasia, which is not really a coral, but it's used often in symbiosis studies for corals, just because it's much easier to keep in a lab setting and they have faster generation times than corals do. But what intrigued me and what I've been trying to do for the past several years is to develop Ostrangia into a model system, just like you have mice or fruit flies in the lab. And why would I think that Ostrangia might make a good model system? Well, with these pictures, it gives you one sense. So they make a skeleton, a calcium carbonate skeleton, just like tropical corals. They have a symbiotic relationship with dinoflagellates, just like tropical corals. But unlike tropical corals, they can survive being in a bleach state here. You see these colonies or these polyps that are completely transparent. So it has a facultative symbiosis with those algae. And so that really gives us a great advantage and opens up a wide range of studies that we can do in the lab. And that clear tissue also helps us see through the tissue and what's happening at the skeleton. The other really remarkable thing about Ostrangia is its distribution. So it's here in Woods Hole up here, but it's its very northernmost limit. And it actually is found all the way down the East Coast, all the way down the East Coast into the Caribbean. And so that spans over 30 degrees of latitude. It's amazing. So it has this really large distribution. But if you think about during the wintertime, it also experiences within the population that lives here in Woods Hole or in New England is this incredible change in temperature. So it goes from, now it can be maybe 23 up to 25 degrees Celsius, but in the winter it goes down to 0 degrees. And I can tell you that's not very comfortable. and most tropical corals, when they get stressed and bleach, it's only because of a temperature change of just a few degrees Celsius. So what makes this coral so tough and be able to withstand these wide ranges of temperature? So I'm hoping that it will give us clues to find out. So we find it all over here. So the Woods Hole Oceanographic Institution is right across the street from us, and they have a dock where all their big ocean-going vessels come in. And covering those pilings are lots of colonies of Ostrangia, you can see here in the middle. So we've started doing some population studies just to better understand what their interactions are with other organisms, where they're found, how they survive through the seasons. But they're also really, really hardy, and probably because of that temperature range. So we can keep them in the lab, even in these little cups on the counter, and do a lot of different studies, as I mentioned. So we also keep them at different temperatures in the lab year round. We have them at obviously ambient temperatures. We have running seawater here. And so whatever temperature it is outside in the sound is what we have here in the lab. But we also keep them at 10 degrees, 20 degrees Celsius, and a balmy 25 degrees Celsius. And they do perfectly fine in all of those. And actually, it seems like when you move them into the 25 degrees Celsius, they almost stretch out and say, this feels great. And so then we can use that same molecular approach in terms of understanding calcification to add that added layer of temperature and symbiosis that we can't do with tropical corals. And so we can look at gene expression at those different temperatures in both the aposymbiotic and symbiotic corals, and then under different temperature stresses as well. And then one of the great advantages of being here at the MBL is that unlike a lot of marine labs that, and I should back up and say I still put together a lot of things with chewing gum and baling wire, but we are unique in that we're also a really world-class center of imaging and microscopy, and a lot of really amazing microscopes have been developed right here at the MBL. And so we've been able to take advantage of that and use just regular bright field and fluorescence, but also confocal imaging, SEM, polarization, and light sheets. And I'll go through some of these for you in a minute. And it lets us get at different aspects of both the symbiosis and calcification. So the light sheet is a system that was designed by a collaborator at the University of Essex, Philippe Lassou, and it's called the Large Selective Plane Illuminator. And what this system allows you to do is most imaging, especially if you're using lasers like in a confocal, that laser produces a lot of energy, and you can actually burn and damage your sample, especially one that's photosensitive like corals and their symbionts. So what it does, it spreads the light into a very thin sheet and then steps through the tissue. So that tissue that it just imaged has a chance to rest as it goes through, but then it gives you this three-dimensional image of a live coral in real time. And the lasers allow you to take advantage of both natural fluorescence and other fluorescent dyes to follow things like pH, et cetera. And what we've been using it for mainly is we want to look at or follow the bleaching process. What happens to those dinoflagellates when they bleach? Where do they go? Do they break down, et cetera? And then also follow calcification, both in a healthy coral and then in a coral that's stressed. And the image that you're seeing here is a close-up of the edge of the calyx of the skeleton. So this is all the skeleton. But what's remarkable, and I'll come back to this in a second, is you see all these little squiggly lines in here. And what those represent are different filamentous algae, fungi, cyanobacteria, et cetera, that are all living with the coral inside the skeleton. And so we've also been using this to better understand these endolithic communities and how they grow and change and are impacting the skeleton. And they can also potentially be functioning as in a sort of relationship, potentially symbiotic relationship with the coral itself and exchanging nutrients. We've also been testing on bleaching and reinfection of these corals. So as I said, they can be in a aposymbiotic or symbiotic state, but we want to be able to expel a certain type of symbiont and then reinfect them or re-inject them with a different type of symbiont that may be more or less tolerant to different temperatures. And so it's a really nice system for that, because it is a rather large polyp, and we can use certain techniques like tricking it into a cold shock by giving it menthol. And if we follow the number of symbionts over time, we can see that we can get the number of symbionts down to zero, or almost zero, I should say. And then we can borrow some techniques of microinjection for things like CRISPR-Cas9 that they do a lot here at the MBL, and use that to re-inject those symbionts back into the coral. And so this is just a short clip of an injection with fluorescent dye, fluorescein. Oh, I'm sorry. There we go. So we have a microinjection needle into the mouth of the coral, and they actually kind of grab like they're feeding on it. And in this image, you can see where we're actually, instead of the dye, we're injecting the dinoflagellates, what you see fluorescing here in red. And this is with the light sheet microscope. So you can see the polyp is happy, it's out, and receiving those new symbionts. And so we can test different types of symbionts and follow that over time and see that we can actually infect them. And what we're working on is developing tags for those different symbiont types so that we can follow exactly what happens to them over time when we expose them to different temperatures later on. And we can follow their calcification during this time as well, and compare between the, aposymbiotic and symbiotic colonies at different temperatures. Again, this is something that we can't do at all in tropical corals, because they would die even going between 20 and 27 degrees Celsius. So we're able to see these really interesting patterns where maybe at colder temperatures, the symbionts are actually more parasites than symbionts, and are actually, can make a negative contribution to the overall energy of the coral. And so they have lower calcification rates. And then another thing that we can do here using a different type of imaging is we can actually watch calcification happening in real time. Now, the image that you're seeing now is just a bright field image, but looking underneath a colony that's laying down new skeleton. And so you can see here individual crystals of calcium carbonate. And this is some of those filamentous algae that already, even as it's just making its new skeleton, it's already there. So I think these things play a much bigger role than we've understood so far, instead of focusing just on the dinoflagellates, that these algae could play an important role as well. But what we do have that's really amazing and allows us to not only just see the individual crystals, but really understand the shape of those crystals and how that changes over time as the crystals are growing. And that's through this polychromatic phase contrast microscope that was built by Michael Schreback, another researcher who's based here at the MBL. And so this image, the colors that you see are based on the angle of the crystal face. And I have a short movie here that you can see. And again, this isn't a live coral, this is live tissue. And these are the new skeleton that's laying down, or crystals. And if you watch some of these crystals here, you'll see that they are moving and the color changes over time. It's a little hard to see in this image, but you kind of have to focus on a couple spots where you see actual skeleton growing over time. And so this is just in the course of a few hours. And again, the coral is alive, and we're hoping we can get really fine scale calcification measurements or growth measurements under these different conditions in the lab. And plus they're just really beautiful to look at. So that imaging with the corals and the light sheet and the polyscope really highlighted the need to better understand that ecosystem that's living inside of the skeleton. So we use some SEM techniques to not only look at this overall skeletal structure, but then look at what's living inside the skeleton. So what you're seeing here is some of the epithelial tissue of the coral that's all ciliated. And then this space over here was where the skeleton used to be, and that we dissolved away for this preparation. And you see, it's almost more other things and algae than it is calcium carbonate. And this is just a little bit closer view to see what it is. And we really know very little, or if anything, about this whole ecosystem. So we relied on some of the sequencing that we've done on this to identify some of those species. And we found a literal zoo of organisms that are living in, on, under, around these Ostrangia here. And so 33 different species, multiple phyla across several kingdoms, fungi, different algae, amoeba, other microbes. It's just, it's crazy. And this is an image of a live coral, and you can see this red is again the dinoflagellate that's living in the coral. Here's some of the tentacles, and the mouth would be here. This is the edge of the skeleton, and then these are all just algal cells that are living underneath the live coral on top of the skeleton. And here I just wanted to show you briefly some of the molecular data that we've had and comparing, say, the symbiotic and aposymbiotic colonies. So these columns looking at gene expression in the symbiotic colonies, and these represent the aposymbiotic colonies. And just to see we're able to see really big differences in what genes are being used and turned on or turned off between these different species. And then we can see that same kind of pattern with different temperatures. So we're hoping that Ostrangia will be able to point us to some key proteins that help this particular coral go through bleaching and these temperature changes. So, you know, just to summarize, this coral is, I think, will be a great model system for trying to understand all these different issues that are going on with tropical corals. I mean, it is itself a tropical coral, but it has that facultative symbiosis with the dinoflagellates. It has a really broad temperature range. It can reproduce in the lab. It has really hardy larvae that can be kept in the lab up to 3 months. It has transparent tissue with little or no autofluorescence. It regenerates rapidly. It has this really diverse ecosystem that is associated with it, and there are genetic tools available. So in terms of, you know, the next steps for making it a model system, we can have it reproduce in the lab and produce lots of eggs and sperm, and get lots of swimming larvae from that. And here's one of the larvae from Ostrangia. And they're about 10 microns. They're really, really small. They don't have their symbionts with them at this stage. They get those sometime between the larval stage and when they settle. And that's where the bottleneck is. we can see them starting to settle in the lab, but we haven't been able to close that life cycle completely. And so in order to use some of those CRISPR-Cas9 tools, we need to be able to have the larvae grow up into at least a young adult stage. So this year actually is a time where we're going to be focusing on ways that we can induce them to settle, because they do actually settle in our marine seawater system here. But when we say, okay, we would like you to settle on this plate, they say, no, thanks. But so then how can we actually help reefs? What can we do to help improve their water quality, et cetera, and protect them from damage and from turning from a beautiful, diverse reef into something that's bleaching and potentially dying or dead reef? Well, seaweed farming. So we go back to kelps and seaweed. And I think seaweeds have a lot of benefits, and they can also make a really significant contribution to the UN Sustainable Development Goals. And so it's, at least in Asia, but more and more in the States, it's a really healthy and sustainable food source. It has a lot of vitamins. It can be used in pharmaceuticals. It can also help with gender equality, as the seaweed workforce, it is largely made up of women, and so allows them to have some independence. It also can be used to produce biofuels and produce a lot of biomass. So instead of using something like corn, we could use seaweed to do that and not use valuable land and other resources. The sector is rapidly growing, so it can provide good jobs and economic development for countries, especially small island nations that need it. And so thereby contributing to the blue economy development and more sustainable practices. We can also use it to produce other bioproducts and including biodegradable packaging. We can, almost more importantly, use it to sequester greenhouse gases, because we know as they photosynthesize, they're absorbing CO2, and also reducing the impacts of acidification. It also can increase biodiversity just because it's providing a structure underwater. And it can also impact life on land through the production of natural fertilizers and biostimulants that are already being used. So one of the other projects I'm working on is developing systems to grow tropical seaweeds at a really large scale. And we're doing this through funding by the DOE, RBE Mariner program, and that's their macroalgae research inspiring novel energy resources. So they want to be able to produce biomass for biofuel production that uses no land, no fresh water, and no fertilizer. And as a note, these farms will be the first tropical seaweed farms in US waters, surprisingly enough. And this is just an overview image to give you an idea of a small test farm that we have in Puerto Rico. You can kind of see the lines going out here, and the buoy goes off screen. We're starting first with a red tropical seaweed that is valuable because of the high carrageenan content it has in its tissues, almost 50%. 40 to 50% is made up of carrageenan. And to do this, we have an incredible number of partners and collaborators that span academic institutions, nonprofits, small businesses, large businesses, et cetera. And to do all kinds of things, from just monitoring the farms, how they impact the environment, modeling how they interact with flow and nutrients, understanding how the algae responds to these systems in the environment. And then also we want to be able to transfer the technology that we're developing and outreach to communities to help develop their blue economies in the region. And so where we have our study sites are in 3 locations, one in our near Tampa Bay, Florida, another in Puerto Rico on the South Coast, and one in Belize. Although our main kind of pilot test site is this site here in Puerto Rico. And so basically what we're doing is developing this modular scalable design. And so what you're seeing is just a sky down view of the pilot system that we'll actually put in the water in Puerto Rico. And so it's 33 meters by 116 meters, and it has 32 grow lines on it. And each of these lines will contain the algae. So it's kind of like a hammock. This is the side view. We have these specially designed buoys that will allow us to, we hope, avoid damage from tropical storms that pass through by lowering down to the bottom and waiting the storm out, and then we're able to raise it back up to the surface by just injecting air to the buoy. And so the current state of the technology for seaweed farming is based on, the bulk of the seaweed is grown in Southeast Asia, and it's all basically small farms with lots of manual labor. And this is actually underwater. Here's like a kayak or canoe for scale, another here. And what we want to do is to remove that manual labor and introduce mechanization and make it easier, faster, and have a better livelihood for someone who works on a seaweed farm. So this is a farm system that we currently have in the water right now in Kodiak, Alaska, that's growing sugar kelp, actually. And so it's basically about twice the size of the test farm that we hope to deploy in Puerto Rico. Although we want to, in the following year, we want to try a system like this there as well. And so what we're hoping that these will allow us to do in the tropics is to improve water quality. And we know part of a problem with reefs is stress from nutrient inputs from runoff on land and sedimentation, as well as acidification. So some of the things that we'll be monitoring on these farms, they just went into the water this spring, is monitoring that nitrogen cycling, how much carbon they're taking up, and the sedimentation rate, and also looking at what species are associated with the farm. And these images give you just a couple of samples that, even though we really didn't have anything on the farm system growing at the time, the structure itself was already attracting small juvenile fishes and squid. So we're hoping that these then could be strategically deployed in areas where we know there's a lot of extra runoff, or where habitat has been lost. And we know seagrass beds and mangrove habitats are nursery spaces. So our hope is that a seaweed farm will provide that link and allow those habitats to reconnect and flourish. So more to come on that soon. So I'm just going to leave you now with an image of the UPR Marine Lab at Isamagueyes, and a view also of one of the specially designed service vessels that we have for the farm. That's right here. It's a pontoon that has this gantry with a winch that allows us to do all the farm work that we need to do. It's called the Damicella, and that's a Spanish word for damselfish. And we called the vessel that because of the damselfish are farmers and farm a little piece of algae to lay their eggs in. So I think with that, I will end it and just say thank you, and thanks to my collaborators.
That was really great. Thank you. I was like thinking so much when you're like, it's over. I'm like, wait. I'm still thinking about all of this. I mean, I have to say at the very beginning, I was kind of amazed when I was thinking about the range of this species of coral, because everyone always talks about coral conservation and increasing temperature, and no one ever talks about decreasing temperature, which, globally is not happening. But I was thinking, wonder if this species is like coming around the Cape like a lot of other species because
Right.
of the temperature warming, but then it could actually hit some colder spells. So it could happen more in the
Yeah,
future.
yeah,
I don't know if
we've
you
been
see.
looking for
Yeah,
sure, and we haven't found any yet, but definitely have your students there keep their eye out if they see anything
yeah.
that looks remotely like a coral.
So do you think it's the sort of the switch to the parasitic behavior that takes the coral fitness down, that's limiting how far north it can go? Did I hear that?
Potentially, yeah, because we don't know for sure, right? This is like total speculation, but we know that there have been studies on this particular species, and we know that photosynthesis in their symbionts stops at somewhere between 6 and 10 degrees Celsius. And so if they're going to stay alive, they need to get nutrients from the host coral, and the host coral needs to survive itself. And continue to grow, or at least maintain stasis. And certainly what we've seen in the studies that we've done is they often do lose weight during the winter and lose skeleton. And whether that's from starvation because it's having to share resources with the symbionts. There is evidence that they will expel symbionts when it gets cold. And so oftentimes in the spring, they'll be paler than normal, and then late in the summer they'll be this dark brown color as they build up that population again. But we don't know if it's also in the winter time. They do spend a lot of time kind of shivering in their calyx, and maybe that lets the skeleton be more exposed to um, uh, other burrowing organisms that get into the skeleton. And that's
Hmm.
why we see so much of these endolithic community there. I don't know.
Hmm. Interesting. I think you just answered a question from a student who said, do the corals have any anti-parasite defense?
Well, they do, and we've actually seen it within the light microscope. And I have to get a shorter video for that another time maybe. But it's so all corals, they have these mesenterial filaments, and most of the time they come out of the mouth and they use them to fight for territory between different species or other organisms. But in this coral, we've seen it overnight where the filaments come out, and they're just, you see them, they're just sort of feeling the area and testing it, and whether that's their sweeping, because those mesenterial filaments are covered with nematocysts, lots and lots of nematocysts, so they can potentially help protect themselves that way.
Oh, that's cool. Oh, wow, the questions are like flying in right now. A student who's very interested in disease ecology, could you talk about range shifts and new diseases that you might see in these corals?
Oh, that's a really good question. Yeah, and I know there's several labs in the New England area actually that are really interested in the coral microbiome, and it's something that I think we're just beginning to understand and know that basically, this is a CT scan of through a strangia. So this is a strangia coral, but it's living on top of a Crepidula slipper snail shell. And we know that there are all these compartments just outside of the coral. You have certain types of microbes. Inside the tissue, you have other types of microbes. Around the dinoflagellus, you have another group of microbes. In the skeleton, there's others. And then, in the space between the coral and whatever it's growing on, there's also other microbes. So we know that there's these different compartments, and they're likely playing different roles in terms of nitrogen cycling, carbon cycling, and then certainly with disease and prevalence. And I think as temperatures warm, some of those, but right now that are benign components of the microbiome could become more virulent, and maybe why we're seeing things like the stony coral tissue loss disease spread so quickly. They're just under stress, and it causes this switch that, oh, okay, this one microbe in this community is no longer producing what I need, so I'm going to grow and take over the others, the space that that one used to take up. So yeah, it's certainly a big concern.
Here's a really specific question for you. Have you tried HYM 248 peptide to induce settlement in metamorphosis?
Ooh, no, I haven't yet, but we've talked about doing that this summer.
Now, why would that be? Help us who don't know what that is. Like, why would that induce settlement? Do you know?
Oh, there's a bunch of different cues that the larvae take up, and some are based on secretions by microbes. We know, like, some of the things that we do already for settlement of these tropical corals is we leave settlement surfaces out for several months in seawater, so they develop a nice biofilm, and oftentimes they like calcified algae, crustose coralline algae. And so this compound mimics one of those signals, and so tells them, oh, I should metamorphose. Although I think with that particular one, it's metamorphose and not necessarily settle and stick.
Mm-hmm.
But yeah, we want to kind of try everything.
Yeah. How can the Estrangula survive without zooxanthellae?
Yeah, that's a great question. It could be because of all those other algae that are living inside of its skeleton that also photosynthesize and are probably leaking, sugars and other food for the coral. But it may be also too that they have very large polyps, and as you guys know, New England is very productive. So there's a lot of stuff in the water column compared to the tropics. And so it may be just a food source.
What you just said there, when you were talking about it, I was like, oh, in the Gulf of Maine, when we put anything into the water, it biofouls. And I was like, oh, it's like biofouling. When you had that incredible list of communities, I was like, oh, well, how would you differentiate that from having a community? I don't know. There's like a lot of ways you could possibly look at what that is. Is it biofoul, or is it an intentional community that's trying to build? And then I was thinking, oh, I bet temperature would influence the biofouling too. Like,
yep, yeah, I have a student who's working on that project right now, actually, because we thought the same thing. Temperature as well as symbiont state. If it's aposymbiotic, then it has a different signature than a symbiotic coral, and so maybe that community changes somehow. But yeah, I'm with you. I think it really, to me, looks like a whole ecosystem, and that we just haven't explored and studied before how they interact with one another.
That's cool. Switching to the algae work, talk about, anytime we go to a big monoculture, we tend to get into trouble. So can you talk about that, like potential susceptibility to pests or things that happen in the terrestrial systems when we plant all the same thing? Is there concern? Talk about that a little bit.
Yeah, no, that's definitely a good point and definitely a concern. And they're seeing that now in Indonesia and the Philippines, where they grow most of the seaweed, and it's mostly in monoculture. We're starting with one species in particular, but we definitely have plans to grow multiple species, and ideally multiple species at the same time. We're also exploring ways we can design the gear to actually attract certain species, and whether it's algae or not, but again, to make it more of an ecosystem that has multiple trophic levels, and so can also help in terms of disease and those other negative interactions. But that's certainly part of what we'll be studying in the next couple of years.
For the students on the island right now, just out past the mooring field, there's some yellow buoys out there, and that's actually kelp and algae species, actually a mixed species grouping on Japanese longlines that New Hampshire Sea Grant is working on.
Oh, that's great.
So,
That's great. That's for test site. So you can see it from the
oh
lab?
yeah, it's like right on the edge of our mooring field. So
Oh, awesome.
is a great site for the underwater research class to do research in looking at all that comes to
Right?
that area, right? Because it really increases the diversity and structure and stuff in the water. So
Yeah,
we're
that's
really
really cool.
interested in exploring that with Sea Grant and students and stuff. So
Wonderful.
so a little bit happening
Yeah.
here, not on that scale. Yeah, you're
Still.
talking about. No, that's great. All right, well, we have gotten through a lot of questions. Thank you so much for answering all of those. Everyone, please know that we record these talks and we post them on the Shoals live stream page on our website. That's shoalsmarinelaboratory.org. And you can feel free to share that with whomever you'd like. Next Thursday, July 29th, end of July, can't believe it, is Dr. Rashid Sumaila, who's from the Institute of Oceans and Fisheries and the School of Public Policy and Global Affairs at the University of British Columbia. And his research focuses on the intersection of economics and ocean conservation. He applies bioeconomics and marine ecosystem valuation to looking at big global issues around fisheries subsidies to impacts from oil spills and everything in between. So a big economics, biological sort of crossroads there. So that'll be a fun talk next week. All of the information about all of our talks this summer are on our website. Thank you so much, Loretta, for joining us. It was great. Really learned a lot, enjoyed it, and have a great night. Have a good night, everyone.
You too.
Take care.
Thanks, everyone.
Bye.